I'm Jess. I'm a PhD student at the University of Sydney and a recipient of the Paddy Pallin Science Grant. First things first, I acknowledge and pay my respects to the Bunurong of the Eastern Kulin Nation, who are the traditional custodians of the land and sea country where my research took place, and I'd like to recognise their deep connection to the seals that I love. I'd also like to flag that all animal research described in this video was performed to strict standards with animal ethics approval and wildlife research permits. Now, let me introduce you to the Australian fur seal. Our only endemic fur seal species and an important predator in Australian shelf waters. As you can see, these guys are playful and curious, but can be easily startled when resting on land, and will flee into the water. This is a very mild example, but they are known to stampede in extreme cases. My PhD project is looking at the impacts of vessel noise and visitation on Australian fur seals at Seal Rocks in Victoria, the species' largest breeding colony with the smallest vessel exclusion zone during summer, which coincides with breeding and a time when pups are very young and vulnerable. Importantly, there's also a declining trend in pup numbers at Seal Rocks. Quick side note, approach regulations at seal breeding colonies are managed at a state level. In Victoria, there are 6 significant Australian fur seal breeding sites with vessel exclusion zones. From November to February each year, 5 of these have exclusion zones of 100 to 200 metres to ensure the protection of breeding adults and very young pups. Seal Rocks is the exception, with a 60-metre approach limit year-round. To address this issue, I've taken a multidisciplinary approach and have compared all results between summer and autumn-winter. As you can imagine, we expect to see more vessels out at Seal Rocks during summer when the weather is nicer and people are on holidays, as opposed to winter when it's cold and stormy. For simplicity, from now on I'll refer to summer as peak and autumn-winter as off-peak vessel visitation. First, I assessed the behavioural response of seals to low, medium and high levels of airborne boat engine noise using controlled noise exposure experiments. High-level noise was more likely to cause seals to move away from the study area than low or medium-level noise, and resulted in increased vigilance, decreased resting, and decreased nursing. The observed responses were short-lived, but the biological significance of behavioural disruption depends on a number of factors, including how often they're exposed to loud noise and the underlying health and stress level of the affected individual or population. This brings us to the second part, which involved assessing pup health using blood parameters. The peak and off-peak seasons represent 2 different life stages for pups, and this was evident in their blood parameters. Young pups, aged 3 to 6 weeks, were undergoing the very demanding process of erythropoietic or red blood cell and immune system development. This happens at the same time as peak vessel visitation and potential noise disturbance. Older pups, aged 5 to 6 months, that were sampled in the off-peak seasons had passed this critical stage of development. What this means is that the younger pups may have less capacity to deal with additional stresses. The third part assessed stress using stress hormone concentrations in pup serum, the clear liquid in blood, pup fur samples, scat or poo samples from older animals, and neutrophil lymphocyte or NL ratios in pup blood. When a stress response is triggered, stress hormones are released into the blood within minutes, then unbound hormones are excreted, usually within 24 hours in poos, or deposited into fur while it grows, which is over weeks to months. Stress also has an effect on the cells of the immune system. Very simply, neutrophils go up and lymphocytes go down, along with a host of other physiological and behavioural changes. So a higher NL ratio means more stress. These immune cell changes take hours and are therefore not affected by being captured. There was no difference between peak and off-peak seasons in pup serum samples, likely due to us catching them. However, young pups sampled in the peak seasons had higher stress hormone concentrations in their fur than older pups sampled in the off-peak seasons. Young pups also had higher NL ratios than older pups, indicating higher stress. The concentration of stress hormone metabolites in non-pup scat samples was also higher in the peak season, indicating higher stress overall in the colony during summer, when vessel visitation and disturbance was expected to be higher. The fourth and final part of my project involved monitoring and mapping vessel visitation at Seal Rocks using a combination of underwater passive acoustic monitoring and direct visual observations. This confirmed that vessel visitation and the associated noise was highest during summer. It also showed that up to 33% of vessel approaches during the peak season were not following the regulations and getting too close. This means that the seals experience a higher risk of disturbance from visiting vessels when pups are newly born and vulnerable, and when they're already stressed or have less capacity to deal with additional stress. It is my hope that this research will be considered in future evaluations of approach regulations to help protect seal rocks and other sensitive marine sites. I want to extend a huge thank you to the organisations that have provided funding and support, as well as my amazing supervisors and everyone that helped with fieldwork. This work would not have been possible without you.